So good morning or good afternoon, everybody. It is 9 a.m. at the West Coast and 6 p.m. for us in Europe, where I am and our speaker, today's speaker, is located. It's a great pleasure and an honor for me to continue our Global IMMUNOTALKS research lecture series with introducing another excellent, outstanding speaker, Katja Simon. Katja, we can't see your picture yet. You have to turn on your video, but maybe let me continue. So Katja started her career in Berlin with a PhD that she obtained in immunology at the German Rheumatism Center in Berlin. From there, she became postdoc, moved to France. And I heard you have a special affiliation with France because you actually spent your childhood in France, not in Germany. And so she did a postdoc in Marseille at the Center of Immunology. Then you traveled further, you moved to Oxford, did a second PhD, and there you joined the Wetherall Institute of Molecular Medicine. You stayed there for quite a while. You first became lecturer, then very briefly associate professor, and quickly turned to full professor. And I think you stayed there at full professor roughly 8-9 years until very recently when you moved back to the beautiful city of Berlin in 2022, where you became a W3 professor at the Charité, and you are presently a section head at the Max Delbrück Center in Berlin. Now, you have great expertise, and I'd like to say a little bit about it, but actually I want to say too much because it's your turn today to talk. Your expertise is metabolism and autophagy related to the development of hematopoietic stem cells, but also to the development and maintenance of many other immune cells. Key findings from you are, for instance, that I would like to highlight, that ATG7 is, for instance, essential for hematopoietic stem cell maintenance, that autophagy is a critical regulator of CD8 T cell memory, or that the molecule GIMAP6 regulates autophagy, immune competence, inflammation, and this in mice and humans. And I'm quite sure we will hear more along those lines today, but as we usually do, I'd like to start our presentation with asking you a question that is related or that is particularly relevant for younger researchers. And my question is, what is your specific mentoring style? How did you manage to be successful with this? And how did your style change over time and have evolved over time? So thank you very much for joining us.
Yes, hello. Thank you, Dietmar, for a nice introduction. So just to answer that question, is my mentoring style. So this is mentoring PhD students and postdocs in the lab, I expect, and also beyond the stay in my lab. So I was brought up as a scientist by my PhD supervisor, Av Mitchison, who was known to be a sink or swim supervisor. So I often say that I do the same thing, but I have adapted a little bit since the times that was very common in the UK and I think also in the US. But one thing I should say is that Av he was a pillar of immunology, and he was always really present in my life. He died last year, but he has always given me advice and has mentored me throughout my life, and that was really valuable. So I try and do the same thing with my PhD students and postdocs. And one thing that changed is when I had 3 children, then I realized that I was actually a better mentor even to people also in the same situation. So I'm usually mentoring mothers now and women, and I feel very strongly that they need this quite a lot more than others perhaps. But of course I continue to mentor people that have come through my lab and others that are not mothers, obviously. So that's how it changed, also through my own life experience.
Oh, very, very good. Very nice to hear all this. And now I'm all set and stage is yours. So again, thank you very much for joining us, and we look forward to your presentation.
Okay. So I will talk to you today. Can you see it? Is okay? Yeah. About autophagy in immune health and disease. And as you all know now, the autophagy pathway is a major bulk degradation pathway in the cell. It doesn't only degrade proteins such as the proteasome, but it actually degrades all sorts of bulk materials such as organelles and other cytoplasmic material, and delivers it to the lysosome, where we have hydrolyses that work at low pH and that can degrade this material and then recycle it. I'm not going to take you through all these molecules, but this has been known for about 20 years now, that upstream of the autophagy pathway, there are many molecules that start this membrane, this double membrane vesicle, which is the autophagosome, which can then engulf material. And there's signaling complexes. In immunology, in immune cells, often cytokines or toll-like receptors or T-cell receptor, etc., signals for this, but also upstream of autophagy is mTOR and the detection of ATP. So usually when there's not enough nutrients, autophagy gets turned on through this pathway, and then these molecules come in and they help to recruit membrane material to this growing membrane, which we think starts on the ER, and it's called the omegasome because it looks like an omega. And then these molecules are important for the elongation of this autophagosome, and some that I will mention most are ATG5, ATG7, and LC3. They're all important for the elongation into the growing autophagosome. The material is attracted by when it's ubiquitinated, and there's receptors that recognize this ubiquitinated material. And then the closure of the autophagosome happens, maturation, and then fusion with the lysosome to become an autolysosome and recycling of nutrients. Sorry. Our contribution to this field have been manifold, and of course there's many more people that participated in here, but I'm just going to tell you a little bit where I came from and why I'm working on what I'm working now. So basically, we started to knock out autophagy in these different cell types of the immune system, and found a pattern which I'm going to take you throughout my talk. So the pattern is that autophagy is important in differentiation, and this is the immune system is only one example. For example, the red blood cell is another one, or the NKT cell, but also adipocytes, keratinocytes, etc., need autophagy to differentiate. The second theme is the maintenance of long-lived cells. And here, the apex of this hematopoietic tree is the hematopoietic stem cell, and autophagy is very important to maintain this long-lived cell. And furthermore, there's other cells in the immune system which are long-lived, let's say memory B cells or memory T cells, but also the B1 cells that originates already in the fetus from the embryonic sac. And then lastly, not lastly, actually, I'm going to tell you a little bit about a cell extrinsic role. So this means that autophagy can have a role in another cell type and then has an impact on the immune cells. And the last one, which I will also touch upon, is the prevention of aging. So basically, if I summarize this now, I would say that the maintenance of long-lived cells is very obvious. These cells don't divide very often and can't dilute their debris to their daughter cells, and therefore they require autophagy. But also the differentiating cell, as I mentioned before, or also proliferating cells which have a short lifespan, which may not need to get rid of their debris, will require autophagy. And lastly, they're the cells that are important for have a high protein synthesis rate, just such as plasma B cells and senescent cells. Actually, we haven't really worked on plasma B cells. Other groups did that, but also the senescent cells is not quite clear yet how autophagy will pan out here. So autophagy can degrade lots of big bulk material, such as lipid droplets or mitochondria or the Golgi, or even parts of the nucleus, the ER, the ribosomes, but also just huge protein complexes, and that's very well known in the nervous system. And so that's the degradation, and then the provision is what they can provide are free fatty acids, are nucleotides, are amino acids to the cell, and obviously that will change the metabolism. So why do these cells need autophagy? And I've labeled them here, provision versus degradation. So long-lived cells, there is no dilution of debris possible to the daughter cells, so they need to be degrading this. They need to keep a clean cytoplasm because they're so long-lived. they shouldn't have much ROS, because ROS damages macromolecules. They should be reducing their organelle and DNA damage. On the other hand, differentiating cells need to remodel, so things need to be degraded, and then new organelles should be built. But also this requires building blocks and high ATP. Proliferating cells need a certain metabolism for which autophagy can be helpful, and they need a lot of ATP and building blocks, but the damage does not matter, it's short-lived. The things that are not labeled, autophagy also plays a role, but not in these two categories. And cells with high protein synthesis rate obviously need amino acids and ATP, for which autophagy is really important, that you need to degrade the unwanted protein, so you're keeping the stress to the cell low. So you can imagine that autophagy plays a role in many cell types and under many circumstances. In addition to function, location, and interaction with other cells, this is just a way of kind of showing the immune system in another way. These are also types of immune cells, but not T cells, B cells, etc. So I show you some examples of how autophagy is important in differentiating in T effector cells with a high proliferation rate. So as a virus enters in the body, we have an expansion of the T cells that is called the effector phase, which is a very highly proliferative phase. And as the virus is being fought off, then this effector pool will die down, and a memory pool will establish itself. Well, in the absence of autophagy, this early phase is already quite compromised. So these are looking at, this is a TCR transgenic system where we're looking at percentage of T cells that recognize the antigen. And if you look at it in percentage-wise, without autophagy, that proliferative phase is really compromised. And if you look at that in vitro, if you stimulate the cells with their cognate antigen and look at proliferation, so this is looking at number of divisions with a dye, you see that the wild type proliferates very nicely, but the autophagy, the T cells that haven't got autophagy, so this is a CD4 Cre ATG7 negative cell, they don't proliferate so well on day 3, but eventually they catch up, and on day 10 they look exactly like ATG7, like wild type. So the question is, which autophagy cargo is important for CD4 T cell proliferation? Why do you need autophagy for this? So we decided to look at to make a transgenic mouse, because we were interested in many cell types, and this was just one question where we really wanted to find out the autophagy cargo in vivo. And this is a proximity labeling model that lots of cell biologists have used in vitro in cell lines. Here we hooked this Apex proximity labeling molecule enzyme onto LC3B, which is expressed on the inside and the outside of the autophagosome. Then we cleave off the LC3B on the outside, and what we're left with is an enzyme, proximity labeling enzyme, that if you add biotin, it will biotinylate all the molecules that are inside the autophagosome. And you can harvest that, pull that down with streptavidin, and do proteomics. So we knocked in Apex2 into the endogenous LC3 locus in this. And what we found that in proliferating T cells, we actually found a number of molecules. One was actually P62 here, which I didn't label, which is a receptor, and which is something that we ought to find in the autophagosome. So we were quite pleased about that. But we also found a molecule that's the IL-7 receptor. And as you know, the IL-7 receptor is really highly expressed in naive cells. But as naive cells become effector cells, this gets the surface expression of the IL-7 receptor is reduced. To cut a long story short, what happens is that IL-2 is either can bind to the IL-2 receptor or the IL-7 receptor with a common gamma chain, and then it can either signal for proliferation. But if that's not true. Sorry, the IL-2 can bind to the IL-2 receptor plus the IL-2, the two chains, but this gamma common chain is actually shared between the IL-2 receptor and the IL-7 receptor. So when there's too much IL-7 receptor at the surface, then the gamma common chain is stolen away from the IL-2 receptor, and then proliferation doesn't happen. And you can see this here if you knock out ATG7 and you give the cells IL-2, the downstream pathway, signaling pathway, phosphorylated STAT5, is very much reduced in these cells. And also, I'm not showing you all of it, but it's very clear that in the knockout for ATG7, you have much more IL-7 receptor, which actually is then steals away that gamma common chain. And furthermore, we could co-localize gamma common chain and IL-7 receptor, and it was more co-localized in the ATG7 knockout, which is what's the prediction of our hypothesis. So basically, in the absence of ATG7, IL-7 receptor, in the autophagy degrades the IL-7 receptor, and in the absence of autophagy, there's too much IL-7 receptor on the surface, and that gamma common chain is then no longer binding to the IL-2 receptor, but it's binding to the IL-7 receptor, and then the proliferation doesn't take place. So this is just one example for a differentiating cell. Here's some other examples, which I'm just going to summarize in a cartoon, but I think they are quite different and therefore quite interesting. So one is about erythrocyte. This was actually my first paper in autophagy. So you know that erythroblasts have a big nucleus, and then they have lots of mitochondria here. This is just before they become an erythrocyte, either in the bone marrow, but also sometimes you find them in the blood. And through mitophagy, so the nucleus is expelled, as you know that mature red blood cells don't have a nucleus anymore. But not many people had actually thought about the organelles in there. And it turns out that since autophagy was discovered about 25 years ago or so now, it gets degraded by mitophagy. And this is something that we found, that if you have autophagy, you just end up with these empty sacs of hemoglobin, essentially, which are the red blood cells. But when we knocked out ATG7, the mitochondria started to look a little bit dodgy and unhealthy. They couldn't really get this. Autophagosome was trying to form, here's a double membrane, trying to form around these two mitochondria, but actually couldn't close without ATG7. And therefore, the red blood cells retained a lot of their organelles, and the mice actually died of anemia. So this is a pretty severe phenotype, and it's pretty important that you get, and it's a very nice example of how a differentiating cells remodels. So mitochondria are no longer needed, so they get degraded. This was found by Monica Mortensen, who was my first PhD student. The other interesting role of autophagy in differentiation is in the differentiation of neutrophils. So neutrophils differentiates from this mononuclear progenitor into this multilobulated neutrophil. And it is a very energy-consuming process. And what we found is that neutrophils, like almost all cells, have lipid droplets in which they can store free fatty acids or lipids, so that they can use those when it's needed for example, for their TCA cycle. And autophagy helps to degrade these lipid droplets and retrieve free fatty acids from these, and that can then help to fuel the TCA cycle, which provides the ATP for this differentiation. So there's, and I'll tell you a little bit more about lipolysis and lipophagy in a minute. So one little story I would like to tell you too is about the cell extrinsic role of autophagy, how autophagy controls inflammation in vivo. So we after this lipid droplet finding that we thought, well, actually adipocytes are the major lipid droplets. They have really big lipid droplets, and they store, of course, lipids in our body to then provide it to other cells as free fatty acids when energy is needed. So we thought that maybe lipophagy would be really quite important in that process. And because we're immunologists, we wanted to look at something where immunology was a major playing a role. So basically, I've told you that there's a cell intrinsic function for autophagy, but here we're thinking about cell extrinsic. And cell extrinsic function of autophagy was already kind of described for tumor cells, where amino acids were given to tumor cells from one cell to another, to the tumor cell. but also cytokine secretion is sometimes delivered through autophagy. And more recently and more controversially, mitochondria can be transferred from one cell type to another, and it's thought that perhaps autophagy plays a role here too. So we wanted to address this. So basically, as you know, adipocytes provide free fatty acids to the body, as I've just said. And what you may not know is that there's a very classical lipolysis pathway where all the molecules have been identified. And this gives rise to, in the cell, to free fatty acid and glycerol, which can then be exported to other cells when they're needed. But this is classical lipolysis. But also lipophagy may play a role in this. So we just thought that perhaps for those cells that live in the fat tissue, and there's quite a few immune cells known to be living in the fat tissue, for example, memory T cells or Tregs or others, we know that they often survive by beta oxidation. So we thought that perhaps they live in that environment because they like free fatty acids for their metabolism, and that possibly adipocyte autophagy may play a role here. So Felix, who did this work, decided to work on this particular project and looked at intestinal inflammation, where, for example, these M1, M2 macrophages are very crucial. Not only that, but also intestinal inflammation, or in IBD, actually was found to have 2 GWAS hits in the autophagy pathway that predisposes patients to develop this disease. And furthermore, it is known that where the inflammation happens, the bowel is actually surrounded by creeping fat. So it is possible that that creeping fat either promotes or inhibits inflammation, and that possibly this was linked, the GWAS hits and the creeping fat. So he, first of all, looked at a model of colitis, which is the dextran sulfate, a DSS model, and looked at whether autophagy was induced by DSS, so measured autophagic flux in the mesenteric white adipose tissue, and found that DSS was actually inducing autophagy quite a lot in this tissue. And then he wanted to ask the question whether adipocyte autophagy controls lipolysis, and indeed in tissue explants of adipose tissue, he found that the free fatty acids that were released from this adipose tissue were reduced in the absence of autophagy. This is an adipocyte specific ATG7 knockout. But also in the mice themselves, serum free fatty acids were also reduced, particularly when the mice were stimulated, were induced for colitis with DSS. So apparently, not only lipolysis plays a role here, but also lipophagy can contribute to free fatty acids in some maybe minor way. We still think that in adipocytes, probably the classical lipolysis is the most important one. But we thought that perhaps under inflammatory conditions, maybe lipophagy would have a major role here. So we induced the deletion of ATG7 in our inducible model. You have to make it inducible, otherwise the adipocytes doesn't fully differentiate, as I just told you. So you have to make that knockout in the fully differentiated model, and in the adult. And so what we saw was actually that the colon was a little bit shorter, and that's a sure sign of more inflammation. And when we looked at histology slides, the mice were indeed having a little bit more of a colitic score. And as you can see, the DSS model is hugely variable, even in the wild type. So we had to do quite a lot of mice to see that difference. But it came up consistently and was also in line with pro-inflammatory cytokines that are elevated in these mice, and other clues that inflammation was actually increased in the absence of autophagy. So we thought that, okay, so perhaps this is right in our hypothesis that lipophagy provides free fatty acids that dampen down inflammation via their role in Tregs and in M2 macrophages, all anti-inflammatory. Perhaps this is how this was working. And unfortunately for Felix, or fortunately, we realized that that wasn't the case, because when we actually knocked out the gene that does most of the lipolysis, or provides most of the free fatty acids in that cell, we could see no difference. So it wasn't actually the free fatty acids that was making their inflammation worse. That was very disappointing and took him a lot of time to figure out. And it was actually with the help of the Techna group that we figured that out. So we had to go back to the drawing board and actually then did a lot of omics to find out what was it that was causing that, how autophagy was limiting inflammation here. And what he found is that very few genes transcriptionally changed in the macrophages, but also in the adipose tissue. And the ones that changed in the adipose tissue were enzymes that were making epoxy fatty acids. These are fatty acids. It's a kind of the family of the prostaglandins that are lipid-based signaling molecules, are mostly made in white and brown adipose tissue, but also in the kidney and liver, and have been shown to have effects on other cell types, such as macrophages. And so these are controlled by a transcription factor called NRF2 and enzymes called EPHX. And he found that in the absence of autophagy, NRF2 is increased. That was known before. And NRF2 actually controls these EPHX molecules to make these epoxy fatty acids. And he also found that other target genes of NRF2 were increased in the knockout here in blue. So he found that the adipocytes weren't exactly using autophagy to promote lipophagy and providing free fatty acids, but actually autophagy was important in controlling this antioxidative pathway by making, again, lipid-based signaling molecules. So if you look at this graph here, where we did lipidomics for these particular lipid-based molecules, you can see that the knockout has got far fewer of these than the wild type. And I can't show you all the data, but basically what it does is, in the absence of autophagy, NRF2 is turned on, EPHX1 is upregulated, and this enzyme will turn an anti-inflammatory epoxy fatty acids into a pro-inflammatory fatty acids. And that will make the macrophage produce IL-10. And that's also what we measured at IL-10. And when we inhibited this pathway, we could see that IL-10 was changed. So just to summarize this is, I've just all said this, but there's an underappreciated fat-gut crosstalk in the regulation of inflammation. There's a novel cell extrinsic autophagy-dependent lipid signaling pathway that regulates inflammation. And now we are kind of interested to find out whether it also controls inflammation. These IBD and other these colitis actually go up with age, and it's quite interesting to find out whether autophagy, and I'll get you to the aging in a bit, may play a role here. Okay, so that was the extrinsic pathway, and then I just want to take you to the last bit of my talk, which is about aging. So just to do a kind of transition from this fat cell extrinsic autophagy pathway to the aging, Hofo in the lab actually measured autophagy in young and in old gonadal fat. And it's one of the tissues where it's really striking how autophagy levels fall. So it's here in this case, it's not only LC31, but also, not only LC32, but also LC31. So LC31 is the upper band, and it becomes, when active, it becomes LC32. And in the presence of BAF-A, you have an increase of this lower band. In the old, you see still a little bit of an increase. The ratio is probably still there, but overall, there's much less of either molecules in there. So, and very interesting and striking observation. But to make the transition a little bit better back to immunology, is that over the years, we always found that autophagic flux declines with age in most immune cells. So here's an experiment from a long time ago where we stimulated macrophages from old and young mice. Old is red, young is blue. And we found it already at unstimulated levels, lower autophagy. And when we stimulate them to their toll-like receptor and cytokine receptors, this is even more striking. And this is counting mean LC3 puncta per cell in the old days. Nowadays, we have a better and easier measurement by FACS, which I'll explain to you in a minute. But you see that if we go across the hematopoietic system, there's a small decrease in hematopoietic stem cells, not much in these progenitors, but quite substantial decrease in mature T and B lymphocytes in the spleen when you measure autophagy between young and old mice. And these mice are very old. They are between 22 months and 24 months. But you can already see a decrease a little bit earlier. So this is very striking and a very kind of also interesting application of our work, because when we knock out ATG5 or ATG7 in the hematopoietic system, or when we knock out hematopoietic stem cells, we see this reduced stemness that's typical of an aging hematopoietic stem cell. We see anemia, which I've already told you about. We see an accumulation of immature myeloid cells. You know, there's a myeloid bias with age, and these myeloids have a reduced function and excess inflammatory cytokines, and there's lymphopenia in all sorts of T and B lymphocytes. So autophagy, and also more interestingly, I guess, there's also an excess of damaged mitochondria, an excess of lipid droplets, all down to what I've just explained to you, and an excess of reactive oxygen species, which is a typical cell biology of an aging cell. So altogether, autophagy prevents aging in the hematopoietic system. And I should take you to the slide, which I probably should have shown you first, which is on immune senescence. So this is exactly what happens. Macrophage numbers go up, but the function goes down. T and B, particularly memory, is very compromised in the aged. But meanwhile, there's chronic inflammation, and we call that inflammaging. We also know that autophagy levels drop with age. So is there a correlation between these? Of course, we have already shown a causal correlation by knocking out autophagy. We find exactly this phenotype of the aged hematopoietic immune system. But we wanted to go a little bit further. So I tell you one more last project here on also about this, about long-lived cells, which are the memory T cells. So Dan in my lab, was a PhD student, he knocked out ATG7 with the CD4 CRE promoter and found that the effector phase was actually in these CD8 cells as opposed to CD4 cells, was actually pretty okay. But when he knocked it out in the memory phase, you see a really severe loss of the memory population. And if you, and this is CD4 CRE, which knocks it out in CD4 and CD8 in the thymic stage. And this is quite similar if you look at 22 to 24 months old mice, which is probably, you know, in our mouse house, how long they really live. Then you see the same phenotype here. Is obviously not quite as stark as knocking it out altogether. So the question was, is this related to one another? Does the old memory response looks like this in response to flu vaccination? And I should have said we're looking at flu-specific CD8 T cells. Does this memory response look like that because you've lost autophagy with age? Another PhD in Hanlin did a similar experiment by looking at B cells without autophagy. And again, the knockout has a very poor memory response upon secondary immunization with this model antigen, which is quite similar to old mice. So the question was, again, is this linked? And so the only thing we could think of to do was to actually try and reverse this by giving an autophagy inducer. So this is how we started to work on a molecule called Spermidine. At the time, this was one of the very few inducers of autophagy. There's not many more. Frank Madeo described it, and he'd shown that it does promote longevity via autophagy. This was in worms and in, flies and other model organisms, and also that subsequently people showed that they have a longer lifespan. There was very little known about there was nothing known about the immune system, but we thought, well, why don't we give this molecule, since he's tried it out in mice, we can give it at the same protocol to our mice and see if we can improve memory responses. So spermidine is an interesting molecule. It's an endogenous metabolite. It's a polyamine made from arginine, and it's also found in food. And I've just put one food type down here, which is fermented soya bean, which is a Japanese delicacy. It's only delicious to the Japanese, I would say. But we can, if we don't like this, you can take up spermidine from wheat germ, from fermented food, from mushrooms, from cheese, etc. And as I said, it's been shown to delay aging in yeast worms in mice. So we gave this to these old mice here and found that the spermidine really improved enormously the memory response. So here we're comparing the old in blue with the brown with spermidine. So it's really done a lot of good to the memory response. And if we give it to mice that haven't got autophagy, we can't see it. So at least this, you can conclude that without autophagy, spermidine doesn't work. And if we give it to mice that are in this B cell, so this is measuring IgG1 antibody titers to this model antigen as before, we can see an improvement of the memory response if we give spermidine to the old. I often get asked what happens to the young, and in fact, we could never see anything in the young. And I tell you why that is, but this might not be true in other organisms, but it was certainly true in mice for this particular response. But it might be also true that in other tissues it's different. But then when we found this, we were very excited and thought, well, couldn't we improve vaccination in the elderly by giving them spermidine? So some of the companies that came and were quite interested in patenting this with us and working with us said, but first you need to find out whether it works in human and what's the pathway downstream. So Frank and others had described some pathways, but we couldn't really see that was the case so there was acetylation, there was epigenetic modification via spermidine. But Hanlin in the literature found in yeast and bacteria that spermidine had a very defined other function, which was lending a moiety part of spermidine to this translation factor EF5A. And by the help of these two enzymes, DHS and DOHH, this translation factor gets activated. It gets so-called hypusinated, which is a post-translational modification. And this post-translational modification means that translation is turned on. So that was known in yeast and bacteria, but nobody had really shown it in mammalian cells. And so first of all, we wanted to show that this was actually doing something to autophagy. So if we stop hypusination by knocking down DOHH with cells from Stefan Balabanov, who helped us all the way through this project, is an expert on this pathway, we find that hypusination gets indeed reduced, and also autophagy is very much reduced from when you still have DOHH. So the dependency of autophagy on this pathway. Secondly, we then, after that, we used GC7, which is a very specific inhibitor of DHS for subsequent experiments. And we found that GC7 can reduce the translation rate. And here we measured it by flow cytometry. It's not quite as profound as cyclohexamide, but over a period of 24 hours, it is quite a strong reduction, general reduction of translation. And secondly, we wanted to know whether there were specific proteins, because people had described specific proteins were inhibited when this translation factor wasn't working. So we did ribosome profiling, which also showed an overall decrease in translation initiation and termination. But also it showed that this translation factor is particularly important when 3 prolines or more than 2 or 1 proline is put together on the ribosome. So it's a polyproline stretch, which are very bulky amino acids, and they need a bit of help for the peptide bonds. So hypusin, basically, the hypusinated EF5A helps to orientate these prolines and make them not stall at the ribosome. And we could find, like others in bacteria and yeast, that found that actually the position relative to PPP, where the stalling was happening without GC7. Sorry, this is not very immunological, but it's very exciting in my opinion. So basically, this translation factor is generally important for translation, but also for translation of polyproline-rich stretches, of which many proteins have such a sequence. But we then went on to look at which proteins were actually affected if we gave GC7 and looked at quantitative proteomics or Silex proteomics, and found a molecule that came repeatedly up in these experiments, which was TFEB. And of course, there are other molecules here that we didn't really follow up, but TFEB, it repeatedly came up, and we love TFEB for the reason that TFEB is actually a master transcription factor of autophagy and lysosomal genes. So as I showed you, there are many proteins are necessary to make an autophagosome. So basically having a control over the translation of the master transcription factor was a very clever way to control autophagy altogether. So this master transcription factor not only transcribes ATG9 and lots of other ATG genes, but also lysosomal genes, and as was found by Andrea Balabio. So basically, the pathway, basically, it shows if you knock out DHS, this molecule in the translation factor, that hypocyanate this ER5A, you lose not only DHS, but you also lose hypocyanation, and you also lose TFEB. So it's all in one pathway. And we also tested whether the 3 prolines in TFEB are necessary for the control of translation by ER5A, and that was also true. So basically, the pathway goes as follows. Spermidine hypocyanates a translation factor called ER5A. This translation factor helps polyproline stretches to be translated. One of the proteins that has a polyproline stretch is TFEB, and that controls autophagy, and that controls lymphocytes. After that, a lot of papers came out about the fact that spermidine in the diet, for example, is important for ER5A hypocyanation and is important for neurons. Dan went on to work with Erica Pierce on ER5A in both macrophages and T cells. It's also important in plants, ER5A, and anti-tumor immunity, etc. I'm not putting all of this here, but there's many more papers now that spermidine is downstream of, ER5A is downstream of spermidine. And in the last Poly-Amine Gordon conference, probably half of the talks were on this. So it's very exciting that this downstream pathway has now been discovered and is available for checking how spermidine works. So I just want to tell that we are now moving into human work. So we had to set up an assay by which we could measure autophagy in primary T cells, for example, or any primary cells. And this is a super assay by which we just use the antibody that works for microscopy. And there's a kit actually available that that recognizes both the free LC31 and the lipidated LC32, which is incorporated into the membrane. But with a mild detergent, you can wash out the free LC31, and then you do an intracellular staining of just what remains in the cell, which is the LC32. And you get a nice shift here, which gets even better if you stop the lysosomal degradation with BAF-A. I may have not said this, but baphyllomycin A is really, or any of the lysosomal inhibitors, is really important to catch the flux or the autophagic flux. So basically, we looked in humans and in vaccine samples of young and old humans. That was an RSV vaccine trial where the old were doing not so well. They didn't really respond so well to the RSV vaccine, even though that's the one target population here, because they should be, they are susceptible to this virus and should get vaccinated. So there is a huge drop again in the autophagic flux also in humans. And if we look in this RSV trial for correlation between autophagy and interferon, you see that the people that do the best autophagy also make the best responses. So this is all very encouraging, and together with the fact that we and others have shown that spermidine goes down with age, and so does hypocyanated ER5A in this data here and our data here, and so does TFEB. we thought we could maybe replenish the spermidine pool in older people, improve autophagy, and improve vaccination responses, particularly memory responses, CD8 memory responses. So I've just said this is all reduced with age. And can we now reintroduce spermidine in humans? So sadly, first of all, I have to declare that I am consulting with Oxford HealthSpan and the John Levitt Longevity Labs, which are both companies that sell spermidine, which you can buy there, but you can buy it from another company too. So we're working together with these companies to check whether that give us some of their products, because it's quite expensive. And now we have done, and it's sad to say that I cannot show you the unpublished data of this trial that we've just conducted and finished. So during the pandemic, we decided that that COVID was, of course, a terrible bug for the elderly, and that the vaccination really had to work well. And so this was also the only vaccine where you could actually get approval from the authorities at the time to go fast ethics. So what we did is we used the fact that in the UK, most of the elderly got their COVID vaccine very early and all at the same time. Then 8 weeks later, when we got the ethics through, we gave them spermidine, 6 milligram per day, which is quite a low dose. This much you can probably take up in food, but apparently that's not enough because it does have an effect. So 6 milligram per day, and that, and then we have a washout period. So we're now looking. What we ideally wanted to do is give spermidine during vaccination, which is what we've done in the mice. But because we were a bit late, we started to do this. And some preliminary experiments in mice had actually shown that it's not so much, if you knock out autophagy when memory is already established, you also lose the CD8 T cells. So it's more, rather than the formation, it's actually the maintenance of these memory cells. So we thought over these 13 weeks, we could possibly see that the waning of the immune response was a little bit slowed down with spermidine. We gave it to 20 volunteers. It's a placebo spermidine double-blind study, 20 and 20, and we just completed it. And we looked at antibodies to spike protein and serum neutralizing antibody, which were both surprisingly significantly upregulated. But also we're looking at, about the mistake here, is immune subpopulation, interferon gamma production, and a lot more by using a large Aurora panel. And we're also looking at our downstream pathway, for which we've all got now flow cytometry readouts. And we're trying to understand why some people respond well to this and some others don't respond, and doing single cell RNA-seq. So that's very exciting and amazing that we could see anything. So we're continuing with this work, and it will be published soon. Just want to summarize all of what I've said. We made a novel transgenic mouse that can directly identify autophagosomal contents. We have found that autophagy can cell extrinsically govern inflammation. And this impact of our work over the last few years has identified a potential therapeutic target to reverse immune cell aging, or in fact, it's identified a metabolite. And so just to summarize again, autophagy is important in long-lived quiescent cells. The example was memory T cells, and it keeps the clean cytoplasm, reduces reactive oxygen species and organelles. It remodels the cell during differentiation, helps to adapt to changes in the environment, provides building blocks, and switches mostly to OXPHOS. We found autophagy in proliferating CD4 T cells degrades surface molecule. And here we haven't really worked that much on it, but this is to come. And I just wanted to thank everyone in my team. I have a small team still in Oxford that are working miraculously without me, or perhaps better without me. And I've built a new team in Berlin over the last year. We're still looking for postdoc and PhD students, but who isn't? And I couldn't really list everyone who contributed, but I think I've put it on the slides, hopefully, for the main people that have contributed. And these are my funders. Thank you very much.
Well, thanks, Katja, for this really excellent and very stimulating talk. I mean, I actually have quite a number of questions, but I will not take advantage of my host privilege here. And I would like to refer everybody from the audience to ask questions that you have to Katja. I'm sure there will be many on the X or former Twitter platform. And with this, I already have to close our session. Thank you very much again, Katja. It was a pleasure to have you. And thanks for you all for joining. And I hope that you join again next week when Jenny Mösberg will be our speaker. So have a nice evening to everybody, or afternoon. See you soon.
Thank you for inviting me. Bye-bye.
Pleasure. Bye-bye.